Swayam Prabha. Digital India. Educated India.
Welcome back to my lecture Industrial Biotechnology. Now today I want to discuss another new fermentation process, that is the glutamic acid fermentation process. Now we know that there are 20 essential amino acids that is very much required for us, because essential amino there are 20 different amino acids that we have in nature. That is permutation combination, they form the different type of protein molecules. So we one protein molecules, that is protein, that one amino acids part of the protein molecule, that is that plays very important role, that is the glutamic acid. Now let me tell you that little bit introduction to this glutamic acid, because glutamic acid is considered as 2-amino pentane acid or 2-amino glutaric acid. It can be either of this way. We can see that it has this amino group. We know all the amino acids, they have the amino group and the carboxylic acid group, plus we have CH2 and COOH that we have. So if you look at the glutamic acid, is the alpha amino acid with the formula of this. Now one thing I want to point out, what is the importance of the amino acids in our day-to-day life, because it has been observed that utilization efficiency of any protein depends on the amino acid composition, because if the amino acids, they are not present in a proper, particularly essential amino acid, they are not present in a proper way, then the utilization efficiency of that particular amino acid reduced to a great extent. Now glutamic acid we use largely in the food item, because it gives some kind of typical flavor to the food. That is so it is chemically, empirically it says that C5H9O4N. this is the empirical formula that we have, and as I told you that it has 2 carboxylic acid group and CH2, NH2 and CH. That is the basic molecular structure is idealized in this form with a 2 carboxylic group and 1 amino acid group. Now the acid can lose 1 proton from second carboxylic acid group to form a conjugate base and simply negative anion glutamate. So this is how it forms the monosodium glutamate, because which is largely marketed. That is, this anion is also responsible for the umami flavor of certain food and use for glutamate flavoring, that is monosodium glutarate. In the abbreviation form we call it MSG. Now that glutamic acid is used as a nutritional supplement for food, because as I told you that if a food has some shortage of kind of amino acid, then we find that if we have well balanced, then utilization efficiency increases to a great extent. Such as processed food like cheese. I have already explained what is cheese. Cheese is a kind of fermented food that is used for the storing the milk, protein and fat. And processed food, we know the processed food, particularly we have several processed food now we have in our market, because whatever food that is available in a particular season, so due to the invention of this canned food for processed food, it is possible to have this particular food throughout the year, because we can preserve this for throughout the year. We have processed milk like this, particularly for military people who work in a very remote area. They use this processed food. And soups, then we have wine, seasoning, yogurt, canned syrup, and crab. These are the different food. We use glutamic acid as a nutritional supplement, as well as it gives some kind of flavor to it. Then in beverage, glutamic acid used as a flavor enhancer in beverage such as soft drinks and wine. Now, use of glutamic acid in health and personal care. Glutamic acid can be used in cosmetics and personal care product, because in day-to-day life several cosmetic products, so it is used for the cosmetic and the personal care product. Glutamic acid can be used as a baby product, bath product, cleansing product, eye makeup, saving preparations, hair and skin care products. Cosmetics and personal care products, glutamic acid function primarily as a hair conditioning agent and a skin conditioning agent. This is nowadays lot of us is very careful about our skin as well as our hair. So glutamic acid plays an important role for keeping the quality of that particular materials. And agriculture and animal feed and poultry, glutamic acid can be used as a nutritional supplement in agricultural animal feed and kind of poultry feed, because I told you that utilization efficiency of the protein greatly increase if you see in any food product that the deficiency of a particular amino acid, if you give it, that utilization efficiency increases to some extent. Particularly if you look at the essential amino acid, that plays very important role. Now, disadvantage of glutamic acid, there are several. High dose causes the sweating, because this is the one disadvantage we have. It causes the high blood pressure. This is another disadvantage we have. The sugar in chocolate, this is particularly for the children. They take the chocolate to a great extent, and the sugar in chocolate provides an excellent substance for growing the bacteria, because this is a amino acid, so bacteria can grow because this is kind of considered as a nitrogen source. Bacteria can grow very easily, which produce acids that causes the tooth decay and fattening. So this is the problem that we have with this chocolate, because chocolate, they have this detrimental effect, maybe due to this particular amino acid, because it helps for the growth of several bacteria. Now, if you look at the history of the glutamic acid, the substance was discovered and identified in the year 1866 by German chemist were the Karl Heinrich Rithensen, that who treated the wheat gluten, for which it was named with sulfuric acid. We all know that wheat contains a lot of gluten, because that is kind of protein that we present in the wheat flour. So you know that so called he identified that gluten present in the, and he treated with this sulfuric acid, and from that he recovered this kind of glutamic acid, and since it is coming from gluten, so it is called glutamic acid, because name has come for that. In 1908, Japanese researcher, that is Akida of Tokyo Imperial University, identified brown crystal left behind after the evaporation of large amount of kombu broth as glutamic acid. The kombu broth, kind of fermented product, that if you evaporate, and after that some brown crystal remain, and they identify this as a glutamic acid. In the late 1950s, the fermentation technology was established and used for the commercial production of glutamic acid, and mostly we target for the production of monosodium glutamate. Now, industrial method for the glutamic acid, both the biochemical as well as chemical methods are available, because since this course deals with the industrial biotechnology, so I shall try to concentrate on the biochemical processes. The industrial method for the glutamic acid can be classified as hydrolysis of protein compounds. As I mentioned, that first identification that done from gluten molecule by the German scientist Karls, and then also it is a microbial fermentation process we can produce, and besides that chemical process also can be used for the production of glutamate. Now, the first let me discuss how the through protein hydrolysis we can produce glutamic acid. Now, this is the acid hydrolysis of protein rich compound. The raw material used in this process are those protein rich in glutamic acid such as gluten, soybean cake and casein. So, these are the material that usually considered as the raw material for the production of glutamic acid. The raw materials is hydrolyzed by concentrated hydrochloric acid in corrosion resistant kettle. We know kettle is kind of either made of glass or rubber line for 5 to 6 hours at 110 degrees centigrade. So, if we heat it in presence of this hydrochloric acid, then this material, the gluten, soybean cake and casein, they get hydrolyzed, and when the protein is completely hydrolyzed, charcoal like humus is removed by filtration, because and then the filtrate is then concentrated in vacuum. That vacuum evaporation we can concentrate the filtrate. Now, acid hydrolysis of protein rich compound, the concentrated syrup liquid is cooled down to -5 degrees centigrade for several hours, and during this process of cooling, the hydrochloric acid salt of L-glutamic acid crystallizes out. So, when you cool down, then in the solution we have already seen in case of citric acid fermentation process also, as your temperature reduces, then the citric acid crystals will be separated out. Here also it is similar to that. When you make a solution of concentrated solution of this hydrolyzed material and cool it down to -5 degrees centigrade, then crystalline of glutamic acid hydrochloride, that will be crystallized out. The crystal glutamic acid is separated from the mother liquid by mechanical means by centrifugation or by filtration. This is kind of physical separation technique we use for the separation of these crystals. The glutamic acid hydrochloride is redissolved in the hot water, and iron is removed from the salt solution, and the solution is then decolorized with sodium sulfite and activated charcoal. We have already pointed out activated charcoal largely used in the industry for the decolorization of the solution, which is largely both is used in chemical and biochemical industries. Now, finally, the crystalline that glutamic acid is obtained by adjusting the pH of the solution with the isoelectric point pH 3.2 and separating the precipitate. This is the finally we can separate out particular this glutamic acid like this. Now, if you look at the steps involved in this particular glutamic acid for that process from protein hydrolysis, so we take gluten, we soybean cake and casein. First we hydrolyze with respect by adding the hydrochloric acid. The hydrolysis take place, you do the filtration to separate the humus or kind of organic material that we have, and then we concentrate it in vacuum, then we cool it down and crystallize, and the crystals we separate it out we get glutamic acid hydrochloride crystals. In the HCl mother liquor, you can make a solution, then add sodium sulfide, and then for removing the iron, then we add activated carbon to remove the color, and then we adjust the pH to 3.2, and then we separate the glutamic acid, the glutamic acid mother liquor, we separate the glutamic acid. That is how it is done. Now let me tell you something about the microbial fermentation process, how it is done. Here the microorganism that is used, Micrococcus glutamicus, this is the kind of microorganism that is used for the glutamic acid production. Now in the culture media, we require different carbon, we require carbon source like glucose or acid hydrolysate of starch. What do you mean by acid hydrolysate of starch? Because when we hydrolyze the starch molecules, so we get oligosaccharide. We not only get only glucose, we get monomer, we have dimer, trimer, or different type of oligosaccharide. So that considered as the acid hydrolysate. Then molasses and a mixture of these substances. Nitrogen source, we use the urea and other chemicals is present. So as per the requirement, we know that for the growth of bacteria, we note carbon source, we note nitrogen source, minerals and vitamins. The prepared media is sterilized in a fermenter by steam, and when the temperature of the media cool down to 30 degrees centigrade, microorganism is added to the fermenter in proper inoculum size. I told you that inoculum size of the industrial fermentation process or normal fermentation process is usually varies from 5 to 10% volume. Now in some cases the volume might be more, so that, but usually the volume of the inoculum should be 5 to 10%. Now microorganism is inoculated 36 to 48 hours, during which the time the pH, temperature and aeration rates are carefully controlled, because this is the aerobic fermentation process. So all these things are and I told you that typical property of the living system, they are very sensitive to environment. So temperature and pH, and because aerobic organism, they can take the oxygen that is dissolved in the media. So this is to be maintained properly so that the growth of the organism takes place. When the fermentation is finished, fermentation broth is hydrolyzed with hydrochloric acid, and glutamic acid is obtained in a process analogous to that for the recovery of protein hydrolysate. So it is that we after the fermentation over, we get the different cell mass and other products, and that is hydrolyzed by using the hydrochloric acid. And we have seen in protein hydrolysis we use the hydrochloric acid, and during this hydrolysis we produce the glutamic acid. Similar to that process is used. Now if you look at the steps involved for this process is like this. We use different, we use the HCl, we use starch hydrolysis, we use the HCl, and so that we get the soluble, where starch usually the insoluble molecules, some soluble starch also there, but that is to be degraded to the monomer or dimer so that your organism can utilize it. So then molasses, then it contains good amount of sugar. We required air because for increase the dissolved oxygen concentration. The culture seed, we required the chemicals. We required as per the required for the organisms, and urea is considered as a nitrogen source. Now when hydrolyze that acid, the first steps that we have the neutralization, because when we hydrolyze with acid, the pH of the solution will decrease to a great extent. So you have to adjust the pH, and to the extent to the pH that is whatever is required for the fermentation process. Then we do the sterilization and treat it with main culture media, because then we mix this together, maybe all these things together, and we sterilize that. We that culture, that chemicals, urea, we mix together, take it to the fermenter, then we sterilize, then we pass air to it, and then we add culture to it. So this is the first we add culture, then we do the aeration. Then we do the pH control as per the requirement of the process. Now after fermentation is over, up to 36 to 48 hours, we get the fermentation broth. Then we concentrate by vacuum, and then we again put the acid for acid hydrolysis. And then here also, like protein, we have seen that gluten and other molecule, that when you do the acid hydrolysis, humus, carbonaceous material like humus formation is taking place. So that humus you separate it out through the filtration process, then filtered, you concentrate, cooled and crystallize, and separation take place, where you can get two type of material after the separation of glutamic acid crystals of hydrochloride. You will get in the crystal form. Also you get a good amount of glutamic acid hydrochloride in a soluble form in the mother liquid. This you can put it back here and concentrate further, so that more glutamic acid can be recovered. And here also, like protein hydrolysis, you have to remove the iron by using sodium sulfide. You have to use the decolorization by using activated charcoal. Then you have to do the neutralization to 3.2, as per similar as we did in case of hydrolysis of the protein. Then separation, we get the glutamic acid. So this is the process through which we can get the glutamic acid. Now if you look at the process flow diagram, how it produces, and actually in practice we have molasses, we have tapioca, or then we have nutrient sources, we have nitrogen, we have air, then we take it to the glutamic acid. This is carried out in a fermenter. This is the aerobic fermentation process. Then we had hydrochloric acid, and this is glutamic acid extractor, then glutamic acid separator. We adjust the pH with caustic soda, then crystallization of the glutamic acid take place, filtration, you dry the crystals, sieving it, and hydrolyze, and then we put it in the plastic bags for storage. And this is how the glutamic acid is produced in the industry, because this is the process flow diagram how it is produced. And if you look at the biochemical pathways, how the glutamic acid production take place, that also very interesting, that we know the TCA cycle, because in the TCA cycle, first you have glucose. When we take the glucose first, both aerobic and anaerobic organism, they pass through the Embden-Mayrov pathway. After Embden-Mayrov pathway, they produce the pyruvic acid, and after pyruvic acid they form the acetyl CoA. Then we have malate, glyoxylate, the isocitrate, citric acid, oxal acetate, acetyl CoA. They in combination they produce the citric acid and this isocitric acid, and then this is from oxal glutarate, and this oxal glutarate, it produces the L-glutamate acid. This is how through the biochemical pathway how it is produced, the glutamic acid, and this is what is in practice in the industry. So this is all about the glutamic acid fermentation process, because I told you that glutamic acid is a amino acid. The amino acid largely used as a food supplement, not only it is used as a flavor enhancer, the several products we use this glutamic acid, and both glutamic acid can be produced by both biological means, by chemical means. Biologically we produce by using the protein hydrolysis as well as microbial fermentation process. When we use the protein hydrolysis, we use the gluten, we use different type of proteinaceous material, we hydrolyze with H2SO4, and I told you that that wheat starch is a good source of gluten, because whatever protein is present that in the wheat starch is the wheat is the gluten. That gluten, when we hydrolyze with HCl, then we glutamic acid we produce, then we separate out this humus from that, and then we concentrate it, and then we crystallize this, and when we do the crystallization, we can separate out the glutamic acid and the mother liquid. Mother liquid also contains the good amount of concentrated glutamic acid. Now similarly, microbial fermentation, the fermentation process also we can use for this is the bacterial fermentation process we use for the glutamic acid fermentation process. Main purpose is to produce the cell mass, and then we dry it and we hydrolyze with HCl. After hydrolysis of HCl, then usually we follow the same process as we use for the protein hydrolysis, and only the thing is that we use the sodium sulfide to remove the iron content in the fermentation broth, and also we use the activated charcoal to remove the color from that. So only I can give you the small thing about the chemical process, that synthetic process that is used by the industry. Since the beginning in 19th century, synthetic methods of producing glutamic acid have been intensely studied by many investigators like Gresham and the Selemoth and Gunthi. They worked together, and they found out, have presented method for producing glutamic acid from various raw material and by synthetic means. But I am not going in details about the chemical process, because main purpose of the course is describe the biotechnological process. So in short, I can tell you that like glutamic acid, we can produce lysine also, and we know that lysine is we stay in a country where rice is a staple food, and rice the protein has a lack of lysine, and lysine, if we supplement with the rice, the protein utilization efficiency of rice increases to a great extent. The protein that particularly amino acids, free amino acids we use in a different form. We use not only by addition with the food products, but we also doctors sometimes recommend in the form of tonic that we use when we have some health problem. They give this because there's some lack of amino acid might be deficiencies there. Then they add some the tonic which contains some kind of amino acid which is required for a particular person. Thank you very much.